as far as clicker scores are concerned, you may notice it's not plugged in. We won't have any today because what would I be asking clicker questions on? So those of you have to leave, bye. But you will probably also notice that many of you will not have scores for your clickers as far as that's concerned. What you need to do is go to course content here in D2L, and then down at the bottom is an iClicker registration. So if you click on that, then it should give you a link right there to link and put in your Odin username and your clicker number. That way I'll get your number. Yeah?
So I don't know if anybody else had this problem, but I couldn't get it to work on Safari. And I was able to figure out how to open up in Chrome.
Yeah.
But I just don't know if that was...
Thank you. I haven't tried. Yeah, so again, for people who are not here, for some reason a few fewer people here today than Wednesday, then try Chrome if it doesn't work with Safari.
Yeah, it wasn't, I was clicking that register button 100 times and
It wasn't doing anything?
Yeah.
So Chrome. Yeah, give Chrome a try. Probably Exploder would work. Okay. Anything else? Okay, let's get started with, who cares about human viruses? Boring. But first, I do have your scantrons here. Yes, they were graded. Again, there's, as I posted on D2L, somewhere there's a ringer in here. I don't know who you are, but I could probably figure out. Got 47. I normally will take that high score and normalize all the other ones to that, but that's clearly an outlier. In fact, there's a 47 and a 45, which were clearly outliers. So I'm probably going to be normalizing to something more like 42. So if you then normalize 90% of that, et cetera, all the way through. So any questions on sort of that grading issue? There were a couple of questions that I wanted to go over in a little bit more detail because obviously I did a really bad job of telling you about them because more than 80% or about 80% of people got them wrong. So this particular one, to do a plaque assay, you need sensitive cells, permissive cells, cell lysis, one-step growth curve, or A and B. Why is it not E? Sensitive, not susceptible. And a couple people asked me during the exam, is that a typo? No, it was not a typo. So that was that one. And that, I think, most people thought sensitive was susceptible. Probably if I were reading the exam, I'd read it as susceptible as well. So the other one, which I think is probably more important, we're going to be talking much more about this as we move on through the rest of the course, has with matrix proteins. And maybe this was confusing in terms of the way the question was worded. But matrix proteins are generally extracellular host membrane proteins, externally exposed viral membrane proteins, internal host membrane proteins, internally exposed viral membrane proteins, or virus receptors. This had about 20% all the way across. People picked A through E. So evidently, I didn't really explain this very well. So the matrix protein, these are the ones that sit on the inside of the viral membrane. So viral envelope, it's a viral protein that then is involved in really holding the membrane proteins, so the envelope proteins of the virus, to everything else. And we'll talk a lot more about this as we move through a lot of the envelope viruses through, and particularly towards the end of the course. And so they're sitting on the inside of the viral membrane, and I was trying to come up with a decent way to describe that, but pretty sure this is the best one here.
When you're talking about that with other biologists, how do you differentiate between virus matrix proteins, cellular matrix proteins, and extracellular matrix proteins? Because there's matrix in all of those. So
Right.
You have to include that.
So
when you say matrix, what gives you the clue?
what gives you the clue here? So when you're talking, okay, maybe I'm just too virocentric here. So the question is, how do you differentiate between all the various different things? Extracellular matrix. You can also think about, particularly for bacteria, like peptidoglycan is kind of a matrix on the outside as well. So yeah, how do you differentiate between that? And so potentially it would have been a viral matrix protein, but the only matrix proteins which we've talked about, or matrix at all in this class, have been these viral matrix proteins. So in terms of discussing scientifically, again, any virologist, when you're talking to each other, will assume a viral matrix protein. And we'll see. A lot of proteins, so we already talked a little bit about the M2 protein from influenza, matrix protein. It also serves as an ion channel. HIV, the matrix protein, is really critical for putting together the HIV particle. Very often these will be what we call M, and you'll see this again and again when we talk about various different viral proteins. The M protein will be the matrix protein. Yeah?
I have a question about the plaque assays.
Yeah.
If it's a lysogenic infection, there will be no plaque, right? Does that mean the cells actually lysoed as a whole?
Okay, so the question is here is, in fact, a really good one, which is having to do with a plaque. And if you have a lysogen, will that actually form a plaque? And now a lysogen, of course, is the host cell. That's the cell that has the virus in it. And so if you are trying to infect a lysogen, if you then induce production of virus there, then yes, that would form a lytic plaque or a clear plaque. And one of the things that unfortunately I wasn't here to mention about lambda is there are really two kinds of plaques. There are clear plaques and there are turbid plaques. A turbid plaque means you have a little bit of growth. It's a lot less than the growth of the cells that are not affected by the virus. And the C1, C2, and C3, anybody get why they're named C1, C2, and C3? Because they're clear, and that means if you have a mutant in C1, C2, or C3, you never get lysogeny. It's always going to be clear. So these are all the clear plaque mutants. So the clear plaque mutants were C1, C2, and C3. And then eventually we figured out what they did. So clear plaques are going to be those that are lytic, but then turbid plaques are then you still have some growth, which is there, and those are the lysogenic or all kinds of other things that could be happening that we haven't really talked about. Our viruses, in fact, make really turbid plaques for most of the part. So, okay, other questions on plaque assays, that kind of thing. Okay, so today we're going to talk about the PicoRNaviruses. A lot of people who work on these call them PicoRNaviruses. Why would you call it a PicoRNavirus? Because you want to have a new name. I think PicoRNA makes much more sense. They're small RNA viruses. We'll talk a little bit about polio. Polio's been in the news, and hopefully it's going to be out of the news in the relatively near future. Hopefully, certainly in our lifetimes, there won't be native polio on Earth, which would be extremely cool if that were the case. Then we'll talk a little bit more about the molecular virology, because this is basically a molecular virology course. A little bit about entry, which we already talked about before a little bit. Viral entry, particularly for polio, is really interesting and different from most animal viruses because it's like bacteriophage. It makes a hole in the membrane. So instead of going receptor-mediated endocytosis, et cetera. One thing that we have not talked about at all, but is really pretty unique to the PicoRNaviruses and then the related viruses, is this whole concept of a polyprotein. And so they just make one big protein and it gets chopped up in terms of how to make the rest of the virus. These are RNA viruses. They've got RNA-dependent RNA polymerases. We'll talk about that. It's really quite well known, and then how that replication takes place. So our sort of standard outline here, where did they come from? Why are people interested? Again, that's mostly going to be polio. The structure, these are pseudo T equals 3 icosahedrally symmetric particles. Binding and entry, we just sort of talked about, makes this pore process. The genome is single-stranded RNA. Interestingly, it also has a protein bound to one end, so it's not completely RNA. There's this protein which is covalently bound to one end of the genome. That has to do with how it replicates. Translation, not so critical here in terms of the actual process of how translation takes place. Much more important is how you go from this polyprotein to all the individual proteins that you end up needing to make the virion over here on the right-hand side. And then finally, how it gets out. This is actually very similar to what happens with bacteriophage. You just get cell lysis that takes place. There are lots and lots and lots of PicoRNaviruses. The probably most confusing term here are the so-called enteroviruses up here at the top, because some of these replicate in the GI tract, but a lot of them are respiratory viruses as well. Polio normally replicates gastrointestinally, but of course the disease you see is when it moves into neurons. These are neurotrophic, of course, meningitis, encephalitis, etc. That's when it gets out of the GI tract. But poliovirus is basically an enteric virus, which is why hygiene is so critical in terms of dealing with polio. Rhinoviruses, probably most of us have rhinoviruses. Rhino is the nose, and so pretty much, I'd say 70 or 80% of common colds are due to these rhinoviruses. And then there are a number of other ones here that they mention. Foot and mouth disease is probably one of the most important animal viruses. And then the first of what's going to be a whole bunch of different hepatitis viruses. This hepatitis A virus is one of these PicoRNaviruses. And in the book it says 4. There's a site called PicoRNaviruses.org. There was actually a list of, I think, 18 genera that were on there. So this is just a few of the PicoRNavirus family. But the main thing that people care about or have studied in terms of PicoRNaviruses is polio. Usually, polio is completely asymptomatic, which is one of the reasons it's really hard to eradicate, because you don't know if you have polio. It's replicating in your gut, goes through, you have basically 0 symptoms whatsoever. But in about 1 in 200 cases, you'll get paralysis, and of those, 5 to 10% of people die, and unfortunately, most of these are kids under 5. And this is really why people got, very interested in trying to get rid of polio. It's by far and away the major cause of paralysis in children throughout the world. And certainly before we had vaccines pretty much everywhere. Developed world, of course, a lot less now. There's a couple of different strains, and we'll get back to this one a bit later. The Sabin strain, which is the main vaccine which is used in most of the world. This is the oral polio vaccine. Turns out it doesn't replicate in neurons, which is why it doesn't cause paralysis, but it replicates really well in the gut. And that has to do with a change in the iris that we'll get to a little bit later on. So what does this look like? This is a really classic presentation of poliomyelitis, which is that neural disease. Can you guys see this? Can I turn the lights out? Maybe we'll turn the lights in front. A little better. So before there was a vaccine, 35,000 kids in the U.S. came down with poliomyelitis, so the neural disease, the paralysis of the legs, per year. Just amazing numbers. So this is really, and this was before 1950, so the population of the U.S. was also considerably lower at the time. Very typical disease phenotype, and there are these limbs which are just not well formed. We've known about what we think is something like polio for over 3,000 years. This is about 4,000-year-old Egyptian carving, now in the glyptothek in Denmark, but certainly looks as if this person has polio, is walking with a crutch, and has this really pretty typical phenotype. So it's been around for a while. Vaccines were originally developed almost exactly 60 years ago. So the 60th anniversary of the original polio vaccine was on April 12th. That was Jonas Salk. He came up with an inactivated polio vaccine, basically taking polio, the virus itself, making huge amounts of it, and then inactivating it. And then using that, and this is also known as the IPV. This is the injected polio vaccine, so polio shots. In 2 years after the introduction of this vaccine, 90% reduction of polio in the U.S. Just an absolutely amazing progress that had happened. And then, polio in the U.S. was gone by 79, just because of this vaccine. And then the Sabin vaccine, which is an attenuated vaccine. And we haven't really talked too much about attenuation yet. The idea of attenuation is you take a virus and you do an infection with this virus. That then produces more virus. You take that virus, you infect, and do this again and again and again, usually in a mouse model, or in this case, they're able to develop cell culture In fact, there's a Nobel Prize that was given for developing cell culture for growing polio. And the more times you do an infection, purify the virus, do an infection, purify the virus, do an infection, purify the virus, what happens is the pathogenicity of that virus goes down. And that makes sense because if you think about it from the virus's point of view, killing off your host is a really bad idea. So the more times you end up, it's really an evolutionary process. You know, people talk about in vitro evolution, you know, doing evolution in a test tube. Well, the vaccinologists have been doing this since Louis Pasteur originally developed this for rabies, which was taking a virus, infecting an organism, getting that virus again, and reinfecting that organism until eventually it's no longer causing disease, but gives you a really nice immune response. And that was developed, the Sabin vaccine, in 1961, so about 6 years later than the Salk vaccine. And there are a couple of reasons why this is the major vaccine of choice, and we'll get back to that a little bit later on. There was this huge progress that happened in the US between 55 and 57, but polio is still endemic in many parts of the world. And so here's some data from polioeradication.org, fabulous website if you're interested in some of these things. Basically a couple of maps. And this is where we had in the winter season, which is sort of the major season usually for polio, August to February 2009, 2010. In 2012, we were down to basically 6 countries. All the dots here represent cases of poliomyelitis. So again, you can't really tell usually if anybody's got polio, it's just replicating in the gut. However, if you have these neurologic disease, then you can tell. And so basically each of these spots represents a kid who is having these particular symptoms. Gone well down from there, and 2014, we're down to, you know, a few extra countries. Interestingly enough, you'll notice these are countries which are not very stable politically. You know, we've got Syria, Iraq, Afghanistan, Pakistan, one case here in Nigeria, and now the latest, which I just pulled up last night from this site here, is Nigeria. There are basically now 3 countries in the world which are endemic for polio, so we're getting really close. In Nigeria in 2013, there were 53 cases. In 2014, there were 6, and only 1 in the second half of 2014. So far, absolutely none have been reported. Now, there's some reporting issues clearly because where these sites are in northern Nigeria is also where there's a lot of activity of a certain group called Boko Haram, so reporting may not be as good as we would like it to be. But the important thing here is really this one red dot. There are a couple of these blue dots on here. If they're blue or green, it's hard to tell. I thought they were blue before. These are vaccine-related outbreaks, and we'll get back to that a little bit later on. But these should not be those that get transmitted. But this one here, the red one, is clearly a wild type normal virus which is being spread. Afghanistan is the second of those places. Only 20 in the second half of 2014, so actually doing really quite well. But if you notice here, almost all these cases are on the border with Pakistan, which is a place where most of wild polio outbreaks are happening, and they're in almost all of them, these tribal regions right here on the border, which again is where there are lots of political issues. 206 in the second half of 2014. It's the only country in the world that is clearly exporting polio to other countries. However, there's been a lot of progress in Pakistan, literally just in the last couple of years, and that has been the public has apparently finally decided that vaccination is a good thing. That was for a long time, there was a huge amount of resistance to vaccination. Vaccinators were getting shot, which was really rather sad. But apparently now there's over 99% approval. This is a recent survey that was done through this university on the East Coast that nobody cares about. And 99% of parents are for vaccination. And a lot of that has to do with getting local people as being the vaccinators, rather than trying to get the guys in their bunny suits and all these nasty Westerners who are trying to sterilize our kids. So that has been hopefully, I think I'd bet a six-pack in the next 10 years. It'll be gone. Yeah.
I wanted to ask about, so I mean, if polio is usually not pathogenic necessarily, vaccinating, we go to where there's cases, like visible cases. Like, is it possible that it will exist and then come back?
That's this slide. Good question. Yeah. So, hang on. Let's go one more. So yeah, what's next? And as again, it's replicating. You have no idea. And it turns out some of you may have heard about there were some cases in Israel last year, I think, where all of a sudden there was, unfortunately, there was no paralysis, but people noticed that there was actually some infectious poliovirus circulating there, and that has to do with this attenuated vaccine. So the attenuated vaccine still replicates. It works just fine in the stomach. And in a very few cases, what happens is you have mutations which then lead to neurotrophic disease. So anytime you've got an actively replicating virus, it can undergo these mutations, and you can get, at very low levels, some spontaneous disease which happens. The big advantage that you have with this attenuated strain is, again, it's replicating in the gut. This attenuated strain now gets left out of the gut, and in particular in places where hygiene may not be the best, you actually end up with this, what they call a halo effect. As people get vaccinated, then are going to be vaccinating everybody else who is around them with this strain, which probably then leads to more possibilities to get the mutations, but also gives you much better spread. So you don't actually have to be immunizing absolutely everyone to get a pretty good community vaccination. The other big advantage here is it's drops, and so really easy to administer. On the other hand, the Salk vaccine is an injection. And anybody who's trying to tell me here that this kid is this happy when they're getting an injection in their leg, I can't believe it. So maybe there's no needle at the end of that. I think that must have been. But one of the things here is there's a lot of discussion about what's going to happen once the wild poliovirus is gone. There are going to be some of this, the oral polio, the attenuated virus, which is still circulating. So how do we need to get rid of that? And that, we're probably going to need to go back to the inactivated, because that doesn't replicate anymore. It's not replicating anymore. It's not going to undergo the mutation to bring back a real one. So one of the things that people are doing right now is ramping up production of this injected polio vaccine, or trying to come up with other ways to deliver this inactivated polio vaccine. So once we have the wild taken care of, again, with the oral polio vaccine, we're going to have to switch over to the inactivated polio vaccine. So that's it for disease and vaccines. Now we can talk about the cool stuff, at least as far as I'm concerned, with these viruses. Starting out with structure, and we've basically used polio already as a nice example of a T equals 3 icosahedrally symmetrical particle, but has 3 different proteins. So anytime you have different proteins, that gives you a pseudo. But they could be 3 different proteins, they could be 2 different proteins, et cetera, which is another one of the questions that we had on the exam. There are 3 proteins, VP1, VP2, and VP3. Again, incredibly creative naming by the virologists. 60 of each of these. And there's also another structural protein, which is VP4, which sits on the inside of this naked icosahedral particle. A little hard to tell here. Again, you've got to find where your 5-fold axis of symmetry is. Where's my pointer? Here's your 5-fold axis of symmetry. There's another 5-fold axis of symmetry. There's a 6-fold axis right here. So you would go 1, 1, and your T equals 3. What do those structural proteins actually look like? Again, we've looked at this before. These are the structures of VP1, VP2, and VP3. The part which is colored is extremely similar for all 3 of these different proteins, but they've all got these different loop structures on the outside. But how you fit your virion together, it's this jelly roll beta barrel structure, one of the really sort of 3 or 4 main structural motifs that seems to be used to putting viruses together. So this fit together, again, it's 3 times 60 of these different proteins to give you a pseudo T equals 3 icosahedrally symmetric particle. So again, that's all stuff we've talked about before, so I went over it pretty quickly. On the other hand, we haven't talked about entry. So as I mentioned before, polio seems to get its genome inside the cell. And when we're talking about entry, we're just talking about getting genome inside the cell in a very similar fashion to a lot of the bacteriophage. So what happens, there's interaction with the virus receptor binding protein, which turns out to also be one of the major capsid proteins, this VP1 protein, that binds to the poliovirus receptor. Interestingly enough, the binding here is not at the very end of the receptor binding protein, which is sort of where you expect it to be. It's down here more towards the bottom, and this is what they call a canyon in the structure. And probably the reason for that, the reason that the receptor binding is a long way down, is so that you can get this capsid protein right next to the membrane. Because once you get it down right next to the membrane, you have a conformational change of the VP4 protein, which, as we said, is sitting right on the inside. That then will make a channel through the membrane of the host, and then your genome enters the cell. Also mentioned, this genomic RNA also has a protein bound at one end. This is the VPG protein that we'll talk much more about when we talk about replication in just a second. The presence of this protein at the end seems to also be important for entry. Seems to be associating with this part of the VP1 protein, and that sort of pulls it into the cytoplasm. What are the receptors? Again, incredibly creatively named receptor is the PVR, the poliovirus receptor. Turns out, of course, it's also involved in other kinds of interactions, but it was originally discovered through poliovirus receptor. The important thing here is not the names of all of these, but the important thing is what their characteristics are, what they look like. Immunoglobulin superfamily, immunoglobulin superfamily, immunoglobulin superfamily, blah, 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 blah. So most of these, again, are interacting with cell surface receptors, which have these immunoglobulin domains. And the rest of these are, again, not really critical. It's not all of them that have that, but most of them are going to have that. So virus receptor interacts with the VP1 protein. Conformational change happens. The genome comes inside the cell. What does that genome look like? Well, this was, again, really kind of surprising when people finally got the sequence of this genome and some clones to, in fact, work with it. Anybody listening to TWIV or checking out TWIV this week in virology? The guy who runs it basically was the one who first got a clone for this particular virus. So what this genome looks like is really interesting because it's almost all one continuous open reading frame. Over 1,000 amino acids all in one open reading frame, which is really kind of bizarre, and we'll talk about this a little bit more later on. There are some non-coding regions, so 5' non-coding region, a shorter 3' non-coding region. There's a poly A tail at the end of the genome, and again, as we mentioned before, there's a protein bound to one end. If you look at what this parts of this, it's called a polyprotein, are, they are the individual pieces that eventually fit together to make your virus capsid and to replicate the rest of the genome. We already mentioned VP2, VP3, and VP1. Those are those major capsid proteins. VP4 sits down in the middle of that. We then also have a number of non-structural proteins. So normally these would be your early genes which are being made, but they're all part of this one big, ginormous polyprotein. These non-structural proteins are, not surprisingly, important for replication. We'll talk about that. That's the 3D protein over here. But this genome clearly has a problem. And that is, okay, how do you get each of these individual proteins? It's a positive strand RNA, so it can be translated just fine with a little exception. How do you start here with this protein at the end? But for all of these things to be functional, they need to be chopped apart. They're not going to be functional as one big polyprotein. They have to be cut into smaller pieces. So one of the most important proteins, and in fact there are two of them here in this genome and many other of the PicoRNAviruses and the related viruses, there are two proteases, viral proteases, which then do the job of cutting the polyprotein into different pieces. And then here in the 3' non-coding region, not surprisingly, is where you have binding for your replicase, because it's an RNA virus. You need an RNA-dependent RNA polymerase. To replicate that, all polymerases are going to go from 5' to 3'. So to start replication, you've got to be over here. You're going to go this way, the other way. For translation, you have to start at this end and go the other way. So what about this strange non-coding region here at the 5' end? Well, just quickly review. Normally, of course, translation happens from a cap. This is cellular translation. Cap messenger RNA. That cap interacts with the cap binding complex, EIF4F, which is composed of all of these other proteins, EF4E, EF4G, EF4A, and that brings the ribosome to the 5' end of your messenger RNA, this whole association process. And then you have translocation along your messenger RNA till you get to the AUG. Then EIF2 hydrolyzes the GBTP, falls apart, large sum of the ribosome comes together, and you get translation. Well, what about these viruses that don't have caps at the end? How do they get translation to take place? Well, they have these things called internal ribosome entry sites, or IRESes. This is a secondary structure which forms. The secondary structure is much more important than the actual sequence of the nucleotides which are there. And that secondary structure provides a place for some of the translational initiation proteins, but much more importantly, the ribosome, to bind to this RNA in the absence of cap. So these are a place that you can get translation to happen in the middle of your eukaryotic RNA. First, people thought this was just viral. Turns out that there are some cellular examples of this as well, where you get translation that starts from the middle of one of your RNAs. So you have the ribosome that associates here, and then you get translation. Turns out in the case of polio and many of these other PicoRNAviruses, what one of the viral proteases does is it cuts the cellular EIF4G protein. The EIF4G protein is sort of the bridge between the cap binding complex and the rest of your translational initiation complex. And if you then have cleavage of EIF4G, that means that the only translation you're going to get is from RNAs that have these structures in them. What do these structures look like? Here's the example for polio. It's not really that critical what all of these things are. Again, it's mostly about the secondary structure. There are few sequences in there that we'll see are going to be important a little bit later on. Not surprisingly, they've got a start codon. Well, start codon is no different here. It's just getting the ribosome there, which is different. There's this semi-conserved sequence, and I love when people talk about these kinds of conserved sequences. It's got a guanine and N. N stands for any of the 4 nucleotides. What's R stand for? It's a purine. So what's that? Purines are the big ones. So adenine or guanine and an adenine. You're going to find these things all over the place. So it's not, terribly unusual. You do have some regions which are also rich in A's and C's. But the main thing here is we've got all of these secondary structures which are going to get the ribosome to this particular AUG in the absence of a cap, in the absence of cap binding. There are actually 4 different kinds of irises, but the 2 major ones, the ones I just showed you, and this other one, and the main difference here with the class 2 irises is that they don't have an AUG as part of the secondary structure. The AUG is a little bit further away from this iris. But the main point here is that they've got secondary structure, They have this GNRA sequence. The only reason people know that this GNRA is important is if you make mutations in it, then it's no longer functional. So, and then these AC-rich sequences, turns out, bind to a cellular, what they call poly-RC binding protein. So anytime you have a run of cytosines, as long as they're RNA, not DNA, this protein will associate with them. We'll see that it turns out that protein is also really important for replication purposes. So it's not just a cellular protein that's important for getting translation to work, but it's also important for getting replication of the viral genome. So get translation, what do you end up with? Again, it's a polyprotein, literally thousands, and in some cases even more than 1,000 amino acids long. So you have translation of this. This ends up getting chopped into smaller pieces by all of these viral proteases. In the case of polio, there's actually an L protease, but most of the PicoRNAviruses are going to have 2 proteases. The most important of those is the 3C protease. So here just shows where you have this protease activity. The fact that you get proteolysis that happens in this polyprotein in the absence of already having is one of these chicken and egg questions. You know, how do you get proteolysis from a protease which is part of your polyprotein if you haven't had proteolysis first to get that protease out? Turns out that these proteases are in, particularly 2A, but to some extent 3C as well, are in parts of this polyprotein that are really floppy. They've got these flexible linkers between each of these different parts of the protein, which then allow them to be active. And so this 2A protease is active as part of the polyprotein. The same thing is true for the 3C protease. So you have translation. First you chop apart your structural proteins from your non-structural proteins, and all of these then get cut up into smaller and smaller pieces. So it's that sequential proteolysis which gives you all the pieces you need. What's really interesting about this is, you know, these viruses actually replicate really quite well, but you end up with the same amount of your structural proteins as you do of all your non-structural proteins, which is really kind of crazy. It's extremely inefficient because, you know, translation, really energetically intensive, but you're just making one big whole thing. And so you end up with way more non-structural proteins than you actually need for this process. And again, it's inefficient, but it seems to work. These are, you know, some of the most common RNA viruses infecting humans and animals. So it works. So we've got our different proteins. How do you make genome? Of course, it's, you know, your positive strand RNA. You've got to make negative strand, and then you've got to make more positive strand in order to package that into the virions to make infectious virions. How does that work? Well, it's RNA. So what does RNA do? I already talked about this for MS2. Secondary structures. So it's all the secondary structures which are really important for getting replication to take place. You already mentioned the poly-C binding protein, which is important for binding to irises. It also binds to this secondary structure at the very 5' end of the virus genome. It's the poly-C binding protein. This is a cellular protein. Everything which is, and this is a cellular protein. Then you also have binding by this 3C. And 3C was what? The protease, together with the 3D protein, which is the viral replicase. So this all binds at the 5' end. The 5' end, you also have 2 conserved uridine residues that are then covalently attached to this VPG protein. VPG is also known as the 3B protein. VPG has a tyrosine, conserved tyrosine. All of these guys that are using these terminal proteins have conserved tyrosines. Why is tyrosine so important? Because it's got an OH. And what does that OH then bind to? Binds to the 5' phosphate of whatever is here. So tyrosine OHs, we'll see this tyrosine OH again and again and again. So VPG, 2 uracils, and then the rest of your genome. You make protein, again, through this IRES-dependent process. You have the 3B protein, which associates with membrane vesicles. It's also a relatively hydrophobic protein. And the reason for this, and we'll see that lots of viruses are replicating together with membranes in these eukaryotic cells. It's probably just to get all of your proteins together, concentrated in one place inside the cell. And you can also get binding there nicely because there are a couple of transmembrane helices right next to this VPG protein. This VPG protein is just made as a protein and then starts out with making 2 U residues from this polymerase, the 3D polymerase, using this CRE sequence here as a template. Once it's got 2 U's that have been made, and this is the really critical and hard part. You got a protein, that tyrosine, you have to put 2 U's on it. Once you got U's on it, you're great. Why? Because U's base pair with what? A's. What's present at the 3' end of your genome? A poly A tail. So once you have these U residues, now you have 2 U's. That is great because you've got a primer, which is your U's, your template, which is your genome. Now you can replicate your way down to the end of the genome. Now you have a negative strand, which is this brown strand, and then you have more VPG that's been made with U's due to 3CD and this CRE sequence. These U's now are going to bind to the 2 A's, which are at the end of the negative strand genome, and replicate your way through negative strand, making a lot more of the positive strand. The reason you have a lot more positive strand is just like the case for MS2. You don't have to worry about ribosomes. You don't have to worry about any of the rest of this translation machinery. The translation machinery is also moving would be moving down this way along the genome of any of your newly replicated RNAs. And there's no translation from the negative strand, so you don't have to worry about any other proteins, any of the ribosomes that are associated with that. What does this job? It's this 3D polymerase. A lot of times people also talk about this as 3D Pol, and I'm sure all of you noticed immediately, wait a minute, that's not an RNA polymerase, it's a DNA polymerase, right? Well, maybe not. So turns out that the high-resolution structure of the 3D polymerase, this is an RNA-dependent RNA polymerase, is a splitting image of DNA-dependent DNA polymerases, which kind of makes sense because it needs a primer and a template. And the difference between RNA and DNA is not really that different. There's sort of the nucleotides, how you're getting base pairs, all of that stuff. So turns out that this polymerase is really much more like a DNA polymerase, or if anything, DNA polymerases are a lot like the RNA-dependent RNA polymerases. We looked at another structure like this before. Anybody remember from the last midterm? Which we've all forgotten because that was on Wednesday. T7 RNA polymerase also has basically this same structure. So this structure seems to be really good at polymerizing DNA, at polymerizing RNA, using an RNA template, or using a DNA template. So this really seems to be the basis for getting polymerization to take place. The DNA-dependent RNA polymerases that we use are ridiculously complex, all these extra subunits and so on and so forth. And so this is very much the basal kind of structure, apparently, for almost all polymerases. And then one of the ways you can tell that this is the case is the right hand. So we've got thumb, fingers grabbing onto your DNA right there in the middle. The palm is where you actually have your active site structure here. So these are, again, classic kinds of polymerases and has this DNA polymerase-like fold. So we've made our proteins. We've replicated our genome. What do we need to do next? Put it all together. So that's the virus assembly process. And again, these structural proteins are made as a precursor protein that then gets chopped up by the viral protease into the smaller pieces. And once it's cut into these smaller pieces, they assemble in a manner actually really similar to what happens with Phi-X174. You get first trimers that assemble, so each of the triangular faces that you have on your pseudo T equals 3 icosahedron, one each of VP1, 2, and 3. Each of those trimers come together as a hexamer. Not hexamer, sorry. We don't have hexamers on here. These are pentamers, excuse me. So 5 of them come together, 5 of these 3 protein subunits. Those pentamers then assemble into a procapsid, again, not at all dissimilar to what happens with Phi-X174. And then you have to go from procapsid to final capsid, here just shown as being the virion. How does that happen? Again, it's a proteolysis event. You may notice here that this is the VP0 protein, not the VP2 and VP4. So VP0 gets assembled here. You have pentamers. These pentamers come together. Hopefully the RNA gets packaged here. It's not clear whether RNA interacts directly with these pentamers or whether it associates with the procapsid. Once you have all this assembly, then you have another proteolysis event, which is when you cut off VP4, again, that's sitting on the inside of your virus. Now this is finally an infectious virion. So sort of why have all of these extra proteolysis events taking place? There are a couple of reasons for that. Of course, you made it as a polyprotein, so it has to be chopped up in terms of being able to get the individual pieces which are going to fit together. But probably much more importantly, proteolysis is pretty much a unidirectional reaction. It's a hydrolysis because you're adding water, and you're now taking a polypeptide chain that was attached and splitting it into 2 pieces. Then entropically, you're never going to be able to put those things back together. And so proteolysis really gives you a unidirectional assembly process, which is really important if you think about viruses. If your viruses are coming together, falling apart, coming together, falling apart, it's going to be a mess. And so having proteolysis as part of that assembly process gives you a real unidirectionality to assembly. And this is also true, again, for Phi-X174, for all of these other ones where you have proteolysis. Proteolysis really gives you a one-way direction. And here, at the very end of virus assembly, is also kind of a quality control. We only are going to get this final proteolysis to make an infectious particle once you're done. Once you put the RNA in, no RNA, no final proteolysis. Once you have the RNA in, you have final proteolysis, and then that virus is going to be able to replicate. It's in fact active. So we have our whole virion. It's infective, or infectious, sorry. What happens to the host? So I already briefly mentioned this already. All the viral proteases. One of the things that viral proteases do is chop EIF4G. What happens if you chop EIF4G? Cap-dependent translation goes to basically 0. And that's what's shown over here on this, the right-hand side of the slide over here. If you look at protein production, and this is protein production detected by incorporation of radioactively labeled sulfur. So anything that's got methionine or cysteine in it is going to be incorporated here. You'll see lots of radioactivity which is being incorporated into lots of different proteins. After 3 to 5 hours of infection, now you've made the viral proteases. They chop up all of the proteins which don't have irises in them, and the only protein or proteins you're making are going to be the viral proteins, which have just been chopped up from your viral polyprotein. So if you look at virus production here, let's say protein production, high virus infection, your protein production goes way down, and then you have protein production, which is almost all viral protein production. Turns out there's also an inhibition of the RNA polymerase, like we had in the case of the bacteriophage T7. You shut down the cellular DNA-dependent RNA polymerase. Why? So I'm hearing various mutterings here. Because you don't need the cellular DNA-dependent RNA polymerase. You have your own RNA-dependent RNA polymerase, the 3D protein. And so any ribonucleotides that are being used by the cellular processes, who needs those? We don't need those anymore. We just need our stuff. And then the final thing that's involved in host interactions is blocking vesicle transport. Why would polio want to block vesicle transport? Again, totally over-anthropomorphizing here. But where is it replicating? It's replicating on vesicles. If vesicles are being transported, the vesicles are going to be transient. You're going to be going from wherever those vesicles are being produced, the Golgi, ER, et cetera, it's going to be released from the cell. If you want to have a good place to be able to replicate, and lots of different places to replicate inside the cell, then you're going to want to stop vesicles from getting to whatever their targets are. And so there's also some proteins which are involved in that process. So that's all about polio and polio replication. I want to talk about a few other things as far as polio is concerned. The first one is, in fact, something we mentioned, I think it was the first lecture, that the chemical formula for polio is C 332,652, H 492,000, etc. And the reason that we have this is because Eckhard Wimmer's lab made polio from scratch. So they ordered online oligonucleotides, put them together, and made polio. They first showed how they could take just the genome of polio and from that genome make infectious polio particles. And then 11 years later, they were able to do this process of getting all these oligonucleotides and showing that the poliovirus they made was exactly from all these pieces of DNA, which they stitched together. You stitch together these pieces of DNA, you have an RNA polymerase. I'll give you one guess what RNA polymerase they used. T7 RNA polymerase, exactly. And then you have to hook up the VPG protein at one end, and then assemble together with all these proteins, and you actually get infectious virus. So there's a big discussion, and that's actually mostly here in this link here, having to do with what's called dual-use research. And so this is clearly something, again, we're trying to eliminate polio, right? So almost gone in the whole world, but you can order the pieces and put them together and get it to work. Now, it took a really good polio virus research lab 11 years to do this. Now, probably be a lot faster if someone else wanted to do it, but it's another reason why we still have to have these vaccines around and available so that you don't have to worry about this kind of process. But one of the issues was, well, why should you have done this experiment in the first place? Should you even make a whole infectious virus? And the answer, at least that Eckhard Wimmer gave, was that A, you want to know how to do it, if any you ever need to, but also being able to put together a complete and functional virus genome allows you to make all kinds of different changes and know exactly where they are, and be able to make these changes into something that might actually be the next generation vaccine. So one of the things that I mentioned really briefly, because I didn't edit the slide enough, the oral polio vaccine, how do they make it? Through all of these passages through cell culture and animals, etc. We now know what the changes are in the genome for that. We can put those changes into a new genome and make potentially a really good new, and maybe even in some cases it'll be an oral polio vaccine that doesn't revert. This has way more mutations. It turns out that the oral polio vaccine, which is used right now, only has about 12 nucleotide changes in the whole genome. We can make a whole bunch more than that. And it turns out that this particular one here, yes, it's infectious. It's about 1,000-fold less infectious than the wild type because of some of the changes that they put into the genome. So this, in fact, would probably be a really good vaccine stream. So, you know, what do you do about this? There's some interesting ethical questions about whether you should be doing this or not. Mentioned right at the beginning, there are a whole bunch of rhinoviruses that are also these PicoRNAviruses. There were 99 serotypes sequenced recently. All of them have these IRES sequences. What's curious is they're slightly different from the poliovirus that we talked about, because instead of having a poly A tail, they have this hairpin loop structure at the 3' end of the genome. And the question immediately then comes up, well, how do you get replication? How do you get those two U's? It turns out it's not a poly A tail, but there still are a couple of A residues at the very end. It's just not a poly A tail. Poly A tail, by the way, is coded for in the genome of poliovirus. And so if the negative part of the genome is going to have a poly U stretch that then will get copied into the poly A. So it's not poly A polymerase or anything like that happens there. But that hairpin structure seems to basically serve the same role as the poly A tail. What do poly A tails do? They help against degradation. And so here, that hairpin loop structure protects against degradation from that. And this was published a couple of years ago in Science there. The last thing that I want to talk about is some of you may have heard about Enterovirus D68. It's the latest, greatest outbreak of PicoRNAviruses. There was a big outbreak last year, 2014, over 1,000 cases of Enterovirus D68. Causes respiratory illness. Again, one of these really confusing things about enteroviruses. This one has nothing to do with the gut. It's all in the epithelia of the lungs. There's been a lot of discussion about whether this also causes paralysis. There have been, I think, 12 cases of acute paralysis. All of those kids, in fact, recovered, so it's not been a lasting case. Whether it's just guilt by association with this virus or not is not entirely clear. And here's a nice example, an EM, which I pulled from the CDC site. We can see each of these individual virions inside the cell. So also showed you they're nice and small, so they're the pico size. A little hard to see here, but they're also closely associated with vesicles around the outside here. So here's a vesicle where they've been replicating on. Here's another vesicle they've been replicating on as well. So those are our PicoRNAviruses. There are a whole bunch of them. Certainly polio has been around for a while. Almost gone, at least as far as polio is concerned. With a second human viral-caused disease that we've been able to eradicate, make go extinct, depends on your point of view. The pseudo-T structure of irises. Replication is really important in the proteases. We've got this 3D polymerase and how we put it all together. If any of you want your scantrons, they are up here, alphabetical by last name. Have a good weekend, and maybe I'll see you this afternoon at my dad's seminar.